Welcome, everyone. Happy to have you with us this evening. Welcome to the MDI Science Biological Lab Science Cafe. We're excited to get a little closer to some interesting subjects this evening. I know many of you are familiar with some of the beautiful scientific images that come out of these amazing microscopes that scientists use. I know if you're like me, you're often captivated by the vibrancy of these images, but I have absolutely no idea what the scientific story that these images is telling us really is. And so tonight we're really lucky to have three experts in the field of microscopy with us this evening to tell us a little bit more about what's actually happening with these images and how scientists are really learning just fundamental lessons from what these microscopes are teaching us. And so with us this evening, we have Dr. Frederick Bonnet, who is the manager of the MDIBL light microscopy facility. And with him is Dr. Marco Pende, who is a postdoctoral researcher here at the laboratory with Dr. Prayag Murawala. And joining them tonight is Dr. Carissa Tilbury from the University of Maine. And the three of them are actually here on our campus this evening because they are teaching a new course in microscopy to Maine students, so largely graduate, postdoctoral and practicing scientists tonight. So it's a wonderful new opportunity for folks in the state of Maine who want to learn a little bit more about these comprehensive and complex instruments. And so welcome to the three of you. We're super excited to have you with us this evening. And I think, I'm not sure who's going first. I apologize, I should have asked that, but we're okay. Carissa has the honor of being the first. So welcome. We're super excited to see and hear what you have to share with us tonight. So Carissa, welcome.
Right. Well, thank you for that very kind introduction. So I'm going to start out this talk and very quickly hand it off to actually Dr. Bonnet over here. So I'm just going to start this talk with why are we even here? Why do we have a Maine microscopy class? Well, it boils down to the statement on the slide. The microscope with its accessories is by far the least understood, the most inefficiently operated, and the most abused of all laboratory instruments. And that was stated by a man named Charles Shillabur, who actually his life job was training biologists on how to use microscopes. And this was an intro to one of his books. So to avoid this, research institutes actually recruit microscopists to help collaborate with biologists. So I'm going to introduce Frederick Bonnet, who is the light microscopy facility manager.
So, hi everyone. So I'm Frederick. So I manage our microscopy facility here at the institute. And so I will talk about the microscopy facility. So the mission of the light microscopy facility is to provide professional scientific expertise in light microscopy at different levels. So the facility will provide access to a state-of-the-art microscopy and workstation. But not only, the facility will also train newcomers properly. So how to use a microscope, of course, but to help them to get what they need to get for their research. So they will get trained about the theory behind microscopy, so everything which is related to optics and fluorescence, for example. So our microscopy facility, it's open every day, so 24 hours a day, 7 days a week, to all the scientists at MDIBL, of course, but also to all the partners, to the INBRI partner and their collaborators. And also the facility is open to all the summer students who are in our lab, as well as our visiting scientists to help them for their research. So the facility provides basically a service for all these people. And in addition to the research itself, the facility also has a role in the education. So as Jerry said, this week we have the first main microscopy course. So we are teaching microscopy to all the Maine, so all the people in Maine who are interested to learn microscopy. And so the main goal for the facility is basically make sure that our users are successful in their research. So imaging, it's really complex because we have to deal with different problems. So for example, we want to image deep tissue. So this is an issue. Some people want to image really fast. Some people want to image really large volume of data of tissue. And so this Marco will present it. And some people want to image animal behavior. So basically microscopy is a triangle. And so it's volume, its resolution, and its speed. And so we have to find the good imbalance between these three aspects to produce the data that people need. So to illustrate this point, again, the function of the facility will be to provide access to cutting edge hardware and software. So here you have some example. So you have this microscope here, which allow people to take bright field imaging. So in this image, you can see an image of a neuron in a tissue, and you can see this tissue. It's really blurry. So this microscope is nice because it can remove the blur. So unfortunately, on this image, it doesn't work. But you can see the difference between both images. So you will have to believe me on this image. But this microscope helps people to improve the quality of their image. We have also microscope who can acquire really quick image. So for example, in this example, you can see a zebrafish brain, and you can see the calcium which is flashing in the brain. So basically what you are seeing here, it's a fish which is thinking or dreaming, because I don't know if it's sleeping. And then using this microscope, produce really a big file, and then we have to analyze them. So we have also some software which allow us to analyze specific tissue, like you can see here. We can isolate the tissue from the image, and we can quantify this. I will talk about this later during the talk. So in addition to have different systems which cover different applications, we have also to deal with different scales here at the Institute, because we have some lab, like Marco, for example, they work on axolotl, so they need to image really big sample, and we have microscope for this. And on the other side of the spectrum, we have people who need to have high resolution imaging to image really tiny things. And so, for example, here you have an image of C. elegans, and you can see the neuron in the C. elegans with the nuclei. So this is one scale. And then you have also other group at the Institute who want to image inside the nuclei here. So it's the image you have here at the bottom. So we have to cover all this range of animal, and it's what we provide here at the facility.
Frederick,
So yeah?
Sorry, can I interrupt you just for a minute? Your sound is cutting in and out quite a bit, and I'm not sure if there's anything you can do to sort of put the microphone closer. When you're close to the computer, it seems to be better. I don't know. Yeah.
Okay, so
Sorry to interrupt you.
is it better?
Yeah, it's still a little weak, but maybe a little better. Try it.
Okay, so yeah, I apologize for this. It's like with microscopy, there is always an issue somewhere. So that's why we have to troubleshoot all the time. Yeah, so these are the other aspects. So as I said, the time, it's also a problem. So some people need to image really fast. Some people need to image slower, your process. We have the scale, the spatial scale. So as I say, from nanometer to millimeter. And so we need to have all this microscope on the facility to cover all this range of application. And we are using commercial microscope. They are nice, but sometimes they are limited. So that's why some people like Carissa develop their own microscope. And so now she will present her talk.
All right. So here I'm presenting not a microscope that I've actually built, but the microscope one of my colleagues at the Vanderbilt University have built. And what we're seeing here is a multimodal microscope. So you can see what that looks like over here. And this might look very different from what you think a microscope looks like that you may have used in middle school or high school biology lab. In fact, there's no microscope base there at all. So you're like, is she sure that that's a microscope? That in fact really truly is a microscope. And this microscope is really powerful. So what you're seeing over here are some images. And this microscope on a same platform has multiple modalities that are seen here. So I have some nonlinear characteristics up here in A and D that you're seeing. So this is label free. So I didn't have to add any fluorophores. And you're going to see a lot of images that have a lot of tags that are used very widely in biology coming on up. But here what we're looking at is both lipids and proteins in the brain without any labels being added. And here we have some autofluorescence, so cellular metabolism can be seen there, and some collagen structure, which there's not a lot in the brain. And then coupled with that, co-registers, so this red square here, are where these two regions came from. And here we see the thermal image of the brain as in a mouse, as the brain is doing various things while the mouse is asleep. So that's an example of imaging platform with multiple imaging modalities, and really arguably is probably the wave of the future, because our biological questions are becoming multi-parameter, multi-size scales, multi-time scales. Unfortunately, optical techniques can do all of those things. So a little bit more about some other research focused applications that can be used. So I also, as a postdoc at Vanderbilt, worked with a collaborator to try to understand, why do some women go into preterm labor and some women do not? So we used a mouse model for that. And once again, we're looking at a mouse cervix actually here. And all the signatures that you're seeing here were imaged without having any labels added to the image. So we're seeing collagen, we're seeing elastin here in red, and the metabolic profiles of all those cells. Oh, it's not, my mouse is not. Sorry. Oh, wait, here we go. I have the mouse on the screen now. So elastin here in red, kind of describing how much is this tissue going to actually be able to expand and grow and allow the birth process to occur. So over here, this is. Oh.
Carissa, can I just interrupt you for a second? So can you just describe to us a little bit, when you talk about labels? I think there are a lot of folks on tonight that may not even understand, like, why that's necessary, or how you used to do it before this new technology came into play. Can you just tell us a little bit about that?
So labels came from, nature inspired. So scientists had looked at, the first one was really the green fluorescent protein, and it was this annoying thing that people found in jellyfish, and it actually impeded their experimental process. And then they found out that they could use this to their advantage and actually put it into different biological samples to highlight, like, think about it really as a highlighter. If you're reading a book and taking notes, you want to highlight certain things. So the fluorophores are tags that are going to highlight, and you can use different colors and different fluorophores for particular structure features within your biology. So the advantage of not having to do that is you don't have to go through those steps. Maybe you're not introducing something that could be changing your biological system altogether. So in the middle here, here we go. We're actually looking at a living zebrafish underneath a 2-photon microscope in my lab. And here in green, we're looking at the myosin, which is the functional unit of the sarcomere of all muscles that is used to generate the force. And then here in blue is actually FAD. It's a protein that's found, it's an enzyme found in every cell that's involved in many, many metabolic processes. And if you look at this video, you actually see a rupture right there of the myosin in the muscle, because this was a diseased zebrafish with muscular dystrophy. And over here is also a 3D reconstruction model where we have, in this case, we have to use a label for the red, which is the nerve. And then in green here, we're seeing the collagen fibers and understanding how collagen homeostasis, or how collagen is regulated within adipose tissue, to really try to understand how obesity has become such an endemic in the world. So I'm going to pass it off to Frederick, who's going to talk. Marco.
No problem. No problem. Thank you, Carissa. Okay, so welcome to my part of the talk. It deals with advanced histology and systemic microscopy. I will talk a bit about a special technique, which is clearing technique. So not just microscopy per se. I'm inspired by these transparent organisms that exist in nature and transparency, where you can just look through the animals to see, for example, how processes of morphogenesis occur and how individual organs develop. Some transparent animals are already used to study such things. C. elegans or zebrafish are established models, organisms. However, most of the transparent organisms are not used in laboratories. In fact, most model organisms are opaque, usually restricting optical access by microscopes to the outermost surface. When trying to look deep into these specimens, tissue-induced light scattering and optical aberrations quickly blur the view. If we can make these conventional model organisms transparent, we can gain deeper insight in a number of complex biological questions we would all like to study. Oh, so here, yeah, here you can see a flask which is filled with little water bubbles which are surrounded by air. Behind this flask is a picture which you cannot see. The reason why you cannot see this picture is that light travels at different speed in air than it does in water, and thus has a different bending ability in the respective medium. The dimensionless number defining this travel speed we call refractive index. So what is happening when we replace the air content in this flask by water? Suddenly we can see the picture of the gentleman who is regarded as the father of a method we call tissue clearing, Spalteholz. So what did just happen? By replacing the air with water, we suddenly have a medium with the same refractive index in the entire flask, which allows the light to travel at the same speed and thus bend in the same way. The same travel speed of light rays is one of the reasons for transparent objects. Another factor, which is really important for transparency, is how even the surface of an objective is. Here we have, for example, a comparison of frosted versus clear glass. Although frosted and regular glass have the same refractive index, one can see better through the regular glass. The reason for this is that frosted glass has a rough surface, while clear glass has a smooth, polished surface. At the rough surface, we have different light bending effects, such as absorption, diffusion, or reflection. While in clear glass, you have a light transmission. Here you can see a tissue-cleared refractive index matched mouse brain in air. And if I remove the air surrounding the mouse brain and replace it by a refractive index matching medium, then you can see something through the brain. So the condition for transparency of objects are uniform refractive index and a flat, even surface. Sorry. So what is causing tissue to be opaque in animals? And how can we apply what we learned in the previous slides to this? So when you look at cells, you have, for example, water content, which is a refractive index of 1.35. You have lipids with a different refractive index, and you have proteins with even higher refractive index. And by using the same example as in the flask, so what we do is we basically replace the water content of cells with a liquid medium which has a higher refractive index to match the overall refractive index in biological tissue. And so we can make the tissue transparent. With this approach, we can make all kinds of tissues transparent. Now we have these big transparent samples, but how can we analyze them? We can use a special microscopy technique called light jet microscopy. Thereby, a light beam goes through different optical elements which transform the beam into a sheet of light. Let us look at some examples how tissue clearing and light microscopy can be applied to answer biological questions. Here, for example, and how does it work? Here you have a transparent mouse brain, and you basically scan with a sheet of light with the mouse brain. This way you can This is the top view. So you see layer by layer, you create an optical section. And this way, we can make from a 2D a 3D image. Some applications. Sorry. So applications are, we can visualize entire neuronal networks with single cellular resolution. We can even go to subcellular resolution. We can analyze long-range projections in a mouse brain. We can see where the neurons are originating and when they're projecting to. Here you have, for example, a single neuron labeled. So we can show where the neuron is originating and where all the axons are projecting of this neuron. And we can even color code these branches of the neuron. We can also study development. You can see here a mouse brain embryo showing the developing nervous system. You can also study nervous systems of different animals. Here you have a marker for acetylated tubulin, which is basically the developed nervous system, and you can apply this to all kinds of animals. Here we have bristleworms, we have squids, we have zebrafish, and we have the axolotl. We can also label this multiple structures, so not just one. So we have neuropeptides in combination with the nervous system. We can show the proliferative cells in squid. We can show transcription factors, as you can see here in the axolotl limb, or we can show membrane proteins. But we also need to analyze these things. And this is where Frederick comes again.
Thanks, Marco. Hi again. So yeah, we have access to all this microscope, which is nice. We can produce all these images. It's nice. But what we want to do is quantify things. We want to extract value. We want to extract information. And so we have to analyze these images. So here on this tiny movie, you have an example. So you have a 3D reconstruction of a neuron. And then by using software, we can identify properly the nuclei of the neuron and all the neurites. And so as you can see, the computer can recognize this structure. And then from here, we can extract different parameters like neurite branching point, which are in red here. We can quantify the neurite length. We can create volume, and we can extract the volume from the neuron. And we can also extract, for example, the end point of the neuron. So when the neuron finish. So again, if you want to have a look, so the green spots here are the end point. The red sphere are the branching points. This is the origin, so the cell body of the cell, of the neuron. And so you can extract value from this. And so this is the next part. Imaging, it's nice, but we want to extract value. So again, here you have an axolotl limb, which is stained for neuron in green and mitotic cell in orange. And so we put this in a computer, and the computer can, in this case, identify all the neurons, and then it can also identify all the cells which are in proliferation. And so here you have a magnification. And so from here, we can extract value. We can, for example, in this example, we can estimate the distance between the cell which proliferate to the neuron. And here you have a color code based on the distance. So the blue are really close to the neuron, and the red, which are not here on the image, I will take this yellow, are really away from the neuron. And so this is called image analysis. It consists to take your image, extract, segment your cell or your structure, label them, and then you want to extract value to be able to perform quantification and to be able to compare between your sample. And so I think Parisa will finish.
Right. So to wrap us up, we saw a lot of what beautiful microscopy images, and then a lot of what can we quantify off those images. So it takes really a village, right? We hear that about it takes a village to parent. Well, it takes a village to do really wonderful advanced microscopy. And we call that village microscopy makers. And this cartoon illustrates that very nicely. So off on this side of the room, we see the advanced optics, laser photonics, engineers, physicists on this side. And then over on this side is what we view as normal, typical biologists, right? But notice that there's not a wall all the way across this cartoon. And notice also there's a hole in the wall that a box is being passed back and forth through. So in order to make advanced microscopy work well, it really has to be a collaboration between biologists, biomedical scientists, physicists, biomedical engineers, electrical engineers, mechanical engineers. We have to all come together to create the next generation of microscopes, use existing microscopes effectively, and train the next generation. So with that, we're going to end on a really couple pretty pictures that Frederick has taken here at MDIBL on existing microscopes here, and open it up for any questions.
Beautiful image. What can you tell us about these two images that we're looking at?
So yes, so this is a good example. So the image on the left, it's a pig kidney cell that I imaged during a course last year. And so you can see in white the nuclei, you can see in orange the membrane, and you can see in blue some vesicule inside the cell. So this one, I did it. This second image, it's not from me, it's from Anna Sommer, who is working in Jarrod Rawlings' lab. And so you can see muscle from C. elegans in yellow, and in magenta, you can see some protein which are implicated in the translation.
Mm.
And so it's two different, so the image looks the same, but in fact, they come from two different microscopes. This one, it's a wide field microscope, the one I talk about, and this one from a more complex microscope, which is called a spinning disk microscope.
Fascinating. So what I kind of take away from this is you have on one hand this amazing technology, but really without the expertise to interpret, to understand, you know, which scope you need to ask which questions, answer which questions, that's really the challenge. And I think, I am guessing that that's really where you guys come in, because you provide that assistance to folks who are trying to utilize this technology in their work. Can you just tell us a little bit about that, Frederick, how the facility works in terms of helping other scientists interpret this data?
Sure. So as you can see, the microscope, it's complex instrument. It's not just you go to the microscope, put your sample and take an image. So to make sure you are acquiring the good image, you have to understand how the microscope works. So I train people on this. And also to make sure that everything is fine before to let people use a microscope. I discuss with them to make sure the experiment is well designed and we have the good tools to image and to analyze and to interpret it. So it's a lot of discussion. And even if I give access to people to this microscope, basically designing a microscopy experiment requires a lot of coordination between the researcher and the facility to make sure everything is going smooth and nobody lose time by doing the wrong thing with the wrong instrument.
That's important.
Yeah, and so during the course we are doing this week, it's what we try to explain to people. Microscope, it's not easy. You have to understand a lot of things. And if you don't understand how things work, it's easy to create artifact in your image and interpret badly your result.
Yeah, that would be really important. So there are a couple of questions in the chat, Frederick and others.
Yeah, we have a technical issue. So I will stop sharing. I will unplug this.
Is that better? Can you hear me?
Yep, we hear you.
Okay, great. So we have just a couple of questions in the chat, and I see others have their hands raised. One of the questions is asking, how do you become a microscopist? Like, what's your background? How did you sort of transition into this field as opposed to maybe going into bench science? And just wondering if you can tell the audience a little bit about that.
So I'm biologist by training. So I did a PhD in Toulouse, in France. So I have a strong French accent, so that's why I'm French. So I did my PhD in France, in Toulouse, and so I started with working with microscope as a user, so like normal people. And then after my PhD, I didn't know if I wanted to continue in research or in a service. So I wanted to learn more about microscopy. So I did a postdoc in Dresden, in Germany, where they have a really big facility. And here I saw different type of microscope, new microscope that we didn't have in France. And it was a good opportunity to discuss with these people and to see how they support the research. And so I learned a lot, and I decided, no, this is what I want. I prefer to do support. And also, when you work on your project, you stick to one or two microscope and one technique. And as you can see, there is plenty of microscope, plenty of technique. And so if you are in a service, basically you have to play with all this microscope. And so this is what I wanted to do. So after my postdoc, I knew I wanted to do this transition. So I did a small internship on the microscopy facility still in Dresden to learn more from inside this time. And then I saw the position here to lead the facility. And so it was a good opportunity for me to bring this experience from Europe. And so it's what I'm doing here.
That's great. Marco, what about you? How did you come to really specialize in tissue clearing and these techniques?
So my microscopy career actually started as a child. So my grandfather was a professor for immunology and biochemistry in Zagreb. And he gave me a microscope, and I put just all the stuff I could find under a microscope. And I was really fascinated with since I was small. And then at one point, I just decided that I want to be a neuroscientist, and I really liked the different microscope that you can use to analyze neurons. At that time, when I did my master's, I was like fascinated with confocal microscopy. And the moment where I came really to tissue clearing was when I was analyzing a brain slice, and I was a bit frustrated that some sections of this brain slices were cut out, and I could not see the entire picture. And this was in 2013, and at this time point, the clarity paper came out, and where they basically showed the entire brains of mice, and I was, wow, that's really cool. I really want to learn this. And luckily, in my institute in Austria, in Vienna, there was one professor who was actually the first person who combined light sheet microscopy with tissue clearing, like even 5 years before this clarity paper came out. So I knew I wanted to do something with him. So we talked. I did a master's there, then I did my PhD there, and I always loved microscopy. For me, it's partially science, but also partially art. This is why I participate in imaging contests. I like photography, like I do underwater photography. So, you know, when I'm sitting in front of the microscope, I also see beauty in things, not just results.
That's wonderful.
This is how I came to it.
I love it. So I should have mentioned at the get-go that Marco you were the official second place winner of this major microscopy contest this year. Is that right? Was it Zeiss?
No, so Zeiss, I was in the top 12 of 1,300 participants. And I was first in the bioimaging North America. Frederick was
cool.
third. So
wow.
Yeah,
That's
I mean,
exciting.
we push each other a bit, you know. We compare each other's pictures, and then we criticize each other. So we try to push each other.
That's fantastic. That's wonderful. Well, listen, we have lots of questions coming in the chat and a couple raised hands. So I'm just going to move on. Dr. Haller, you've had your hand up for a bit. Do you have a question?
Unmute. Hello, very nice presentations, fancy pictures. When you interact with other scientists, I mean, you had this nice cartoon that you have to work together. How long does it take? I can imagine that you have, solutions within a couple of minutes, but when you interact with other scientists, when you give them advice and then you try something out, is this a continuous effort, or is it solved within, do you need a couple of hours? It's like a doctor's visit. How does it work?
So it depends on kind of what the problem or the scope of the problem is. I will say that in the situations that I find to be the most fun, the most pleasurable, the most successful, it's a continuous discussion, and it's back and forth. And you're really colleagues, right? And you're creating new knowledge together, because there comes a limit, right? So, and the quantification stuff that Frederick had showed, what happens if there's something that you can't quite yet quantify with your existing commercial microscopes? Do you just throw up your hands and say, I'm done? Or is it possible that if you work together, you can overcome the current limits that may have existed, right? And a lot of optical microscopy techniques have done that, right? We have super res that beat the diffraction limit of light, right? And allowed biologists to answer various questions that they never dreamed of being able to answer. So I think realistically, it's an ongoing, continuous, collegial relationship that is the ideal situation.
Yeah, thank you. Thank you very much. That's enlightening. Just one follow-up question. How do you allocate your time? I mean, when I look at these pictures, I would love to spend hours with you guys discussing, how to improve and all that. And your time is limited. So how do you structure that?
Not at all.
Very good.
Just go with the flow.
That's great.
No, you have to time. You have to finish stuff. I mean, you can easy to lose yourself.
Yeah. So in the class today, we actually we're talking about a paper called Reverse Thinking for Experimental Microscopy Design, right? So anytime that you're going to create an image, you should have an objective as to why you're creating that image. But I think that kind of drags you back on track of, I have this mesmerizing image in front of me, but why did I do that again? So you need to quantify that information. And, at the end of the day, it's kind of funds, right? No one's going to give you dollars just to create beautiful images because we're not artists. We are, at the end of the day, scientists that we have to deliver on results. So I think there's a very practical aspect to that as well.
That's important. Right. Dr. Boyer, you've had your hand up. Do you have a question?
Well, I have a comment, then a question. My comment is that it's absolutely amazing that this laboratory, given its size, has these extraordinary state-of-the-art instruments. It's quite an accomplishment, and I think people should be aware of that. The other, my question, it has to do with the studies of the non-tagged tissue, the muscular dystrophy study, where the sarcolemma, the muscles were colored green. Is that the spontaneous fluorescence of the muscle, or is it color-coded by the computer?
So none of the images that you saw are spontaneous color. They're all a color that is added in, and Frederick and Marco are masters at making those colors really pop. So frequently all microscopy images are in grayscale. So it's like a black and white image, and then we add color to them afterwards. So the difference with the green in that particular image is that I didn't need to add anything to that tissue sample to elicit that grayscale value.
Not everybody may know what the difference is between fluorescent light and white light or regular light microscopy. Could you give a brief description of what the difference is?
You're the greatest speaker.
All right, I'll take that one. So white light, if you just have a white light microscope, which is, if you're going to go to the toy store and buy your kid a toy microscope that Marco described getting started out on, you're using either an LED at this point or maybe the sunlight bouncing off a mirror. So that's considered to be white light, right? So frequently that's limited by fairly thin tissue samples. So you're going to either transmit light all the way through, you're going to reflect light back throughout. So anytime light interacts with tissue of any kind, you have reflection, you have transmission, and you have the ability to absorb. So in the term of fluorescence microscopy, you have these special molecules that have different states that once they absorb energy, we're going to actually have an electron get kicked up into a highly energetic state. And then what is called, I like to call it the like photon boogie. So there's a little vibration and rotation, and they lose some energy, and then they emit a photon to go back down to its ground state. And that photon is the fluorescence process of that molecule. So those are a very, very kind of elite group of molecules that exist in nature and that we can engineer based on things that we found in nature, namely the green fluorescent protein found in jellyfish.
Great, thanks.
Great. So Marissa, there are a couple of questions which I think you may have sort of answered that are in the chat, and they're related to color and sort of how you decide. You know, you mentioned that a lot of these images are black and white, and but then, you know, Marco and Frederick are really great at assigning that color. How do you decide? Is it just the natural occurrence, or how do you decide what color is related to which tissue or process?
I'm going to pass that to Frederick.
Yeah, so as Carissa said, we use a fluorophore, and so the fluorophore will produce a certain range of wavelength. Okay, so for example, in the green range, but we record the image in black and white. So if we are imaging the GFP, the green fluorescence protein, we know we are imaging in the green range, but the image is in black and white. So we will color code the pixel in green. If we image the red fluorescence protein, we know the photon will be in the red range of color, but the image will be again black and white. So we will color code the pixel in red. So basically, just this is the rules. If you use a GFP which emits in green, you color code in green. If you use a dye which emits in the blue range, you color code in blue. After, there is rules for this, because as you may know, some people, for example, are colorblind. So if you image green fluorescent protein and red fluorescent protein, naturally, you will color code in green and in red. But you have 10% of the population who cannot see the red or the green, so they will see everything in yellow. So there is some rules when you color code your image. And so, for example, if you want to image green fluorescent protein and red fluorescent protein, you will color code the green in green, but you will color code the red in magenta, which is colorblind friendly. So there is a lot of guideline published by some facility. And so the role of my role, basically, it's to make sure that people stick to this new guideline, because at the end, you want to transmit a message to everyone. And so if 10% of the people you want to talk with cannot see your color, your message is not as efficient as if they can see the color.
Fascinating. I would have never thought of that. That's amazing. So a couple of other questions, Frederick, and others. One is about software. So Alan says he's curious about the imaging software that you use, and he guesses that deblurring and counting things may be old hat, but what about recent application of AI pattern recognition for structure identification and disease diagnosis by cell abnormality or parasite identification?
So sometimes it's a little bit complex, but yes, so we use different software to do the image analysis. And so basically the software will recognize certain value of pixel. This is for segmentation, basically. And to properly isolate your structure of interest from your image, you have to do it manually. You have to play with slider and different options. And so this is time consuming and it's never perfect. So now there is new tools which are based on deep learning. So basically they use the power of the computer to test different possibilities at once. So it's really fast and the results are usually a little bit more accurate than if someone do it manually.
Interesting. So my guess is that's probably an area will be an area of tremendous growth going forward, right? More sophisticated
Exactly. So,
yeah.
So I don't know if we have time, but because we are on my PC, I can show you. I can do a really quick demo if you want. So like this, you can see the power of image analysis. So I will share my screen, and yeah, you should stay because you will see the power of this kind of tool. So I will share my screen. So you see my desktop, and so the software we use, which is free, it's called Image, which is here, for example. So it looks really easy. It's really simple, as you can see. I have to move the bar from this. Okay, and now I will show you an image, which is here. So I open. So you have this nice image, and if you want to segment this image, it will be really difficult because you see you have a lot of blur on your image. And so I will show you, so I will just duplicate. So I have to thank one of our students, Doug Curie, with, during the course this week, because it's one of his samples. So if we want to work with an image like this, basically you will go on image and you will try to segment. So what you would try to do, it's apply a threshold, play with the option. I will not do it here, but you can see there is a lot of area which doesn't segment properly. So in this example, if you want to quantify the neuron, you have a big artifact on your image here, as you can see. So this will not work. So it's where deep learning enter. So I will use a plugin. So I will not enter in the detail, but just watch what I'm doing. I pick an option, which is called lab kit in this example. No, it's fine. So I will open this software, and so it will open my image, as you can see on the screen, right? So the image is a little bit dark, so I will make it brighter, and now you can see the neuron again and the blur, right? So now I will tell the software what is blur and background and what is a neuron. So I will click on this option, background. I will take my pen. I will zoom on the image, if I find the good control. And then I will say, this is my background. This is my background. This blur here, it's my background. And then I will take my foreground and I will say, I will zoom and I will say, this is my foreground. So I want this, I want this, and I want this. Okay. So I just do this couple of steps, and now I will click classification and I will run the algorithm. It will train the algorithm, and then you will see the process in real time.
Wow.
Okay, and now you can see all the part of the image which was completely blurred before. It's not perfect, but still here we will be more accurate than the manual way to do.
Good for a 30-second presentation.
Yeah.
That's great. Thank you.
Oh, Frederick, you go in deep waters.
So this tell you, you see, in 30 seconds, I was able to segment this image, which took several hours to do properly, and it was not as good as the computer.
Yeah, that's
An
amazing.
example of deep learning and deep learning and artificial intelligence.
Fascinating. Marco, there's a question for you here from Juyang about tissue clearing. She says, you know, she can definitely see the upsides of using that technique, but she's curious if there are limitations. In other words, are there any effects by applying the medium to make tissue clearing to normal biology or physiology?
Yeah, so actually tissue clearing is a broad term. You have many different techniques, and some emphasize on the morphology of the tissue. For example, when you have mouse embryos and you do too harsh dehydration or too harsh chemical treatment, then you just destroy the morphology of the sample. Or when you have, for example, a bone and a muscle, and then you do a dehydration-based approach, the dehydration kind of shrinks the sample. The bone is not shrinking, but the muscle is shrinking. So you have kind of an anisotropic deformation of the tissue. And then some approaches are not friendly to RNA, then the RNA gets destroyed. Or sometimes it's very unfriendly to kind of a transgenic signal, which maybe the animal has, like the GFP signal we were talking about. So there are definitely limitations, and like your questions require a certain method. There is not a method which can solve everything. Another thing which you need to be aware is that tissue clearing works in not with live tissue. So you cannot just take an animal which is alive and put it in such a solution. It's too harsh of a chemical treatment. So it's perfect to take snapshots, for example, or just show entire networks, but they're again just snapshots. There are some limitations.
Mm-hmm. Yeah, that makes sense. So, Frederick, I'm guessing this question is for you, but just Hannah asks, given how microscopy has advanced in recent years, what would you say that the limiting factor is now in terms of data? Is it data storage and that kind of thing? What are the big challenges for your field going forward?
Yeah, data storage is one of the big problems because microscope produce more and more data because you can see more and more things. So this is the first issue. And then you have to also process all these images. So if you talk about a file from a light sheet, it's several gigabytes of data or terabytes of data. So you need to have the power to deal with this image because, again, do the imaging, it's not a problem, but you want to extract data. So you want to handle this image, you want to put them on your PC, you want to analyze them with different software. So you should have good power, good computer to do it. And of course, a good storage, because if one experiment saturates the full institute, it's not really good.
And good programs as well.
And good programs.
Yeah, because not every program can handle a certain amount of data. It just crashes. So there's a reason why some programs are really, really expensive. So they allow us to handle actually such data, analyze such data, improve image quality of such data.
Interesting. Marco, we're losing your sound just a little bit, but I wanted to ask you, there's been a bit of news in microscopy here at MDIBL over the last couple of weeks, and you were successful in getting some funding for an exciting new project. Can you tell us just a little bit about that and what will this new microscope bring to this facility?
Yeah, so we will build a light sheet here. It will be a home built system, and it will be kind of the one of three system in the entire US. And it will be perfect for tissue clearing approaches. So we can suddenly analyze here large samples. And you saw some pictures where you could see entire axolotl and entire mouse brain, for example. These were images acquired on a microscope I used in Austria, in Vienna. It was also a home-built system. And by bringing such a system to this area, we will not keep it for ourselves. We will share it with other scientists as well. And this kind of such new methods or microscopes are kind of a beacon for other scientists to come to MDIBL. It should be one of the things which should define our imaging facility.
That's so exciting. So this will be only the third microscope of its kind in the United States. Is that what I heard you say? Yeah, exciting.
There are a couple of light sheet systems. So there are different light sheet systems, but this one specifically is one of the in the US.
Yeah. And
That's
so
exciting.
to continue with what Marco is saying, yes. So we have a nice facility because we have a nice support from Grant and from our president and director, which is really important because we can do this. But we are lucky because it's not the same in all the institutes. So that's why we really put effort to open the facility to everyone. So any scientist, it's in Maine, for example. So the course it's fully funded. So all the applicants are coming on the campus for free to follow this course. And the light sheet, because it's one of the three in the all US, of course, it would be open for all the US, everyone in the US who want to use this system. So also on the facility, I set up a program which allows people who are far away in Maine, for example, because you have to drive a lot, or also outside of Maine to send me the sample and I can image with them remotely through Zoom. Like this, they can have access to our microscope without moving. So without driving, for example, which can be time consuming and you have to leave your family, but they have access to this, to all this microscope and knowledge. And so this is important to mention it. We are completely open and we are looking for more external people.
Excellent. Well, we'll help you get the word out. That's really, really exciting. So thank you all so much for sharing this exciting technology with us, for letting us glimpse some of these beautiful images, and for helping us appreciate sort of the science behind the beauty. And Carissa, I am going to maybe contradict you just a little bit, because I think you guys are artists, and I think what you do is absolutely beautiful, and understanding that there is a purpose behind that. But thank you for sharing that with us and sharing your process. It was really fun to learn a little bit more about the process that you all take when you entertain this work. And I think for me, really fascinating to think about the importance of designing a good experiment and making sure that you really have the right parameters and that you really get it right. Because I can see that it would be pretty easy to get it wrong. So glad to have you all sharing your expertise with others and helping train young scientists with this technology. So thank you for what you do. We really appreciate it. And thanks for sharing it with us this evening. Okay, so this is actually our last Zoom Science Cafe for the summer. We are going to give it a whirl at returning to in-person events. And our next event will be featuring Dr. Herman Haller, who will be introducing a new division of the MDI Biological Laboratory known as MDI Bioscience. So this is something that we launched earlier this year. We're very excited about it. It's an opportunity to really influence early stage drug discovery. So we are going to be utilizing zebrafish embryos and potentially some other organisms to help speed the drug discovery process, to help maybe find some efficiencies in that process. And so if you are interested in learning a bit more about that, I hope you will join us for our next Science Cafe, which you can register online at mdibl.org/events. And I'm just wanting to pull up the date here since I completely just forgot it. So let's see, that is June 27th. So we look forward to seeing you at Marin Auditorium. If you've been with us in the past, you may want to go to the main Center for Biomedical Innovation, but we're going to move our location because it will allow us to have a hybrid option. We have better technology there in the auditorium, so we're going to try it there. So if you're willing to join us in person, we look forward to seeing you, but if not, we will continue to see you online as well. And so thank you all so much for your support of Science Cafes. It's our pleasure to be able to share some of this science with you. So have a great evening. And thanks again to our presenters this evening. It was wonderful.
Thank you.
Bye.
Bye.